Okay, I think we're going to go ahead and get started. My name is Lauren. I'm the Director of Family and Donor Experience at GRIN. In this role, I work closely with families and donors and partners to ensure that the voices of everybody, patients and families impacted by GRIN disorders, are heard and supported. It's an honor to be a part of this journey with such dedicated partners like GRIN2B Foundation, and I'm so excited to be able to collaborate with you guys to present GRIN Therapeutics hosting the Radiprodil trial as a potential treatment for GRIN disorders.
Yes, thank you, Lauren. Yes, I agree. We're very excited. I am Liz Marfia Ash, president of GRIN2B Foundation, a GRIN2B mom, and we are very excited for this clinical trial and this webinar today. And we are very proud to co-host this webinar with our partners at GRIN. And yeah, we're really excited to hear from the team from GRIN Therapeutics. We'll be hearing from Bruce and Hillary and Michael, and then following, we'll have a couple quick questions. And then we'll be hearing from Dr. Kristen Park from the Children's Hospital Colorado. And then at the end, um, we will do a Q&A session at the end.
Awesome.
And I'll
So,
hop
and
in
then
really quickly.
Alyssa, yeah, here. You want to
Yeah, good afternoon, everyone. My name is Alyssa Bustillo, and I am the Executive Director of the GRIN2B Foundation. We were founded in 2017. Our organization was created by parents like Liz seeking answers and support for their children diagnosed with GRIN2B related neurodevelopmental disorder. So since then, we've grown into a global nonprofit organization dedicated to really raising awareness, supporting families, and advancing research for GRIN2B. So over the years, GRIN2B Foundation has funded critical research projects, launched patient support programs, built educational resources for families and clinicians. And we've also established a patient registry to help drive scientific discovery. So we are proud to partner with Cure GRIN to continue these efforts, ensuring that families across the GRIN community really have that support and those resources that they need while we push for really meaningful treatment options. So I'm very excited for this presentation today with GRIN Therapeutics, and I'm really looking forward to the conversation and discussion following the presentation.
Great. Thank you, Alyssa. All right, let me add on the team from GRIN Therapeutics. Bear with me. And one more. Here we go. Okay. I think that's every. Slowly they're coming in. And I will run the slides for you guys. So take it away, Bruce.
Great. Well, thanks very much. It's a pleasure to be here today with you. I'm Bruce Lucher, the CEO of GRIN Therapeutics, and we're very excited. We share the excitement of the broader GRIN community and the GRIN community in particular around the soon-to-be launch of our phase 3 study, studying Radiprodil in GRIN-related neurodevelopmental disorder. And I thought I'd take just a couple of moments to orient you to the journey so far, which has been jam-packed with activity and really encouraging developments and a lot of resilience from the team over here. A lot of that in collaboration with the patient advocacy groups, and in particular, Lauren and Liz, and the collaboration that we've had really since day one. If I step way back and think about the concept mode of where we were about 3.5, 4 years ago on this topic, it was what felt like a wonderful idea to be able to go after the underlying cause of the disease, which is unusual for drugs that are targeting developmental epileptic encephalopathies. Typically, they're trying to carve out symptoms from the disease and treat those symptoms, and certainly that's important. And there has been some success in the space as it relates to the epilepsy in all of these the broader group of developmental epileptic encephalopathies. But we wanted to do more than that when we set out on this journey. We wanted to target the broader disease, and we felt that we had a molecule, a program, a drug candidate that could do that. And for all the right reasons, the biological reasons are pretty clear. We go after the receptor in the disease process that is thought to drive the disorder, and as such, have an opportunity to go after the broader disorder as opposed to cherry picking certain symptoms. We didn't know how much success we would have in our clinical trial, but that was certainly the organizing principle as we kicked this off about 3.5 years ago. And we could not have done it without all of our collaborators, and that it certainly includes the patient advocacy groups with whom we've partnered really since day one. And for those of you who spent time with Hilary Savoie, one of our first employees, it is a commentary on how important we believe the patient advocacy group organization collaboration is to the work we do. And we've continued to push ahead on that, but it hasn't just been the patient advocacy groups, it's been a broader community of individuals who sit around treating these patients. That includes the principal investigators, the key opinion leaders with whom we speak and gather advice and guidance. It includes, of course, the patients and patients' families who've been courageous and brave in progressing with us through this phase 1b study. And it also includes folks who sit on the administrative side of all of this, the international regulators, global regulators, who have also engaged with us in a really constructive way and have us in the position we're in today to be able to pursue what you'll hear as a really creative and promising clinical trial design, different from what other companies have done in the past. And again, that's driven by how specific and targeted our potential treatment is. And we're excited to share that with you. I've talked a little bit about the difference between pursuing symptoms and pursuing the disease. I think the advancements that we've seen around the broader landscape in whole exome sequencing and genetic diagnosis have really enabled a much more targeted and pointed approach to pursuing treatment of these disorders. And we see ourselves on the cutting edge of that kind of treatment approach. There are certainly others who have assumed a similar kind of targeted effort, not in GRIN, but in other DEEs. And we see it as the first step toward disease-specific treatments in the broader DEE space. And that's what you're going to hear a little bit about today as we talk about trial design. And it presents opportunities for patients and families who manage this disease in ways that other programs haven't been able to produce in the past. And then finally, we're here because we have data that give everyone around the table a significant amount of confidence and enthusiasm. As folks may know, we released our top-line data from our Honeycomb study, the phase 1b study in GRIN-related NDD, late last year at a big European epilepsy conference called the ILAE conference. And in that trial, we're able to demonstrate a remarkable, truly remarkable decrease in seizure frequency of 86%. Michael will spend some more time talking about that today, but that 86% median seizure frequency decrease is on top of standard of care. So patients came in on their standing regimen. We observed them, and then we treated them with our experimental therapeutic and saw, even on top of an average of 3 anti-seizure medicines, a profound decrease in seizure frequency, and then saw improvement in non-seizure outcome measures on multiple different kinds of outcome measures. And it was that totality of data that gave us the necessary conviction and motivation to go have the kinds of conversations we've had with regulators, which now put us in a position to initiate our phase 3 study. So quick closing comment for me is just a reiteration of our gratitude and thanks to the community, the broader community of individuals who contribute meaningfully to the broader effort here. And we can't express our gratitude strongly enough. And we look very much forward to maintaining that momentum with you as we head into our phase 3 study. We need it. The program needs it. The disease state needs it. And we're really looking forward to locking arms and partnering with you as we push off here. And with that, I believe that I will turn it over to Dr. Mike Panzera. And Mike, I'll hand it over to you.
Great. Thanks, Bruce. And
Sorry, I wasn't changing the slides.
that's all right. This is actually fine. You're great where you are. This is a perfect place.
Okay.
I'm Mike Panzera. I'm the Chief Medical Officer of Novartis Neurosciences and GRIN Therapeutics. Also, when we get to the Q&A, I'll have Dr. Russell Chin, who's with us also, who's the medical director overseeing this program, and be there. He'll be there to answer questions also about the trial. But first of all, who we are. We're a biotech company based in New York City. We actually have only one funder that has committed to funding us through, hopefully, the approval of the product based on the data. The company was inspired and formed during the 2019 GRIN conference at Emory University. And then this essentially built up this partnership for developing Radiprodil as a potential treatment for GRIN-related disorder and gain-of-function variants. And you'll hear a little bit why Radiprodil would be ideal for this. Bruce already described the top-line data. We'll get into that in a little bit more detail. And throughout the entire process, we've been really working with the community because this is all new. This is new for you. Quite frankly, this is new for all of us. And it's new for the regulatory authorities. They've not had anyone come to them to talk about a disease like GRIN. So it's been a great process of learning together, and you'll see that process has been highly successful as we've continued to move things forward. The next slide, please. So again, why evaluate Radiprodil? Next slide, please. So this is to understand why we want to bring Radiprodil forward. One of the most important things to understand is that what gain and loss of function means and what the NMDA receptor is. This is a protein, like a type of gate, that sits on the cells of the central nervous system, brain and spinal cord. And this gate is there controlling the way that the neurons or the nerve cells communicate with each other, their balance, if you will. And sometimes these gates don't work properly. The gate on these cells either lets too much information flow, or other times not enough, and that's gain of function and loss of function. If you think of the gate as like a gate in a barnyard that's filled with chickens, you can see the illustration on the right where a broken latch on that gate, or a gate that lets too much flow through, chickens flow out. Where it's lost, where the gate is stuck, keeps too much in. And like any other balance, there has to be balance of this inflow and outflow to maintain normality. And in GRIN, with the NMDA receptor, that balance is disrupted, and that leads to the signs and the symptoms that you're all too familiar with. Next slide. So Radiprodil, if you think of Radiprodil as as something that can change that flow, you think that NMDA receptor is encoded by the GRIN genes. And there's a part of that receptor called NR2B or GLUN2B. And these receptors, these proteins, these gates are made up of multiple pieces or subunits. Radiprodil is designed to target one of those subunits, NR2B or GLUN2B. This is sort of one part of that. And it's designed to be what's called a negative allosteric modulator, meaning it's designed to bind to one of those portions of the gate and sort of restore that flow. And the idea here is that when you have a drug like Radiprodil, if you can bind to NR2B in the setting of overactivity, you reduce that overflow, that excess flow, bring it back to approaching a normal flow, and hopefully have fewer seizures and improvement in all the other neurodevelopmental symptoms and behavior. So this is the concept behind why we decided to bring Radiprodil forward to test its effects in GRIN-related neurodevelopmental disorder with gain-of-function variants. Next slide, please. So what we did was design a study called the Honeycomb study. Now, the Honeycomb study was multi-center, meaning there were lots of hospitals involved. It was open label, meaning that everybody got Radiprodil, and it was phase 1B, which means it was an early human study. And it was an add-on study, meaning that we took Radiprodil and added on top of standard of care. So the medicines that people were left in place, and we added Radiprodil on top to see if it could help. We targeted the trial to patients with GRIN1, GRIN2A, 2B, or 2D gain of function variants. The ages we included were from 6 months to 12 years old. And importantly, because of Radiprodil's ability to target the receptor, and that receptor, that NMDA receptor, is important for all signs and symptoms of the disease, whether there are seizures, not seizures, we wanted to be able to study Radiprodil's effect on both types of people living with the disease. Those with what we called countable motor seizures, meaning motor seizures that the physicians and the caregivers could count. Even though they were receiving anti-seizure medications, these events were still occurring. And a behavioral group where people didn't necessarily need to have seizures, but did have behaviors and neurodevelopmental symptoms that were felt to be serious enough that in the setting of a trial, there might be improvement. As with all early studies, the primary endpoint was safety and tolerability. That's very typical of phase 1b studies, and also measuring the drug in the blood, plasma concentrations, to see how we were doing in terms of was the drug moving through the body the way we predicted it to go. The secondary endpoints to that, meaning those which were checked after this, though, were very important and focused on efficacy. So could Radiprodil impact CMS, or countable motor seizure frequency? We looked at whether there could be certain levels of reduction from baseline, or whether there was just an overall change in seizure frequency, as well as a change in seizure-free days, meaning could we increase seizure freedom throughout a given month? We also looked at clinical and caregiver global impression, or CGIC or CAGIC. And again, what we were looking for here was whether there were seizures or whether there were not seizures, were clinicians and caregivers seeing some level of improvement in patients receiving Radiprodil. After 12 patients had completed the first part of the study, we wanted to do a pre-specified analysis to see, was the drug working? We had intended to enroll 12 patients, but we decided to look halfway through to see if there was any benefit. And in the end, there were about 15 patients available at the time we did the initial safety assessment. Next slide, please. This shows the design of the study. What you're seeing is that when patients were first identified, there was a screening period, and that's the period where clinicians are assessing, is a patient who wants to get into the study, will they qualify for the study? That's also the period when the seizures were beginning to be counted to see if the person with the seizures has enough seizures to qualify for the seizure cohort, or whether if they qualify otherwise with the GRIN, they could go in the non-seizure cohort. So that first period, called screening, very typical of a clinical trial, and it's really that period where you have to sign consent, and that's the period that determines if you're eligible for the study. Then there was a titration period, and that's the period where we started with a low dose of Radiprodil, and then every 10 days or so, the dose was gently increased to make sure it was well tolerated. And this occurred over a period where we were targeting a certain level of drug in the blood that has been associated with efficacy in animal models. So we wanted to get to a level that was predicted to have benefit. Once that level was achieved in the blood, we then maintained patients at that dose level for a period of 8 weeks, and then looked to see if there was an effect, and then patients could continue on open label treatment indefinitely. Right now, the study is ongoing, and patients who completed the study are continuing to receive Radiprodil. And the way this is administered, this is a suspension that's administered either orally or via feeding tube twice a day. Next slide. So what we saw in the study was very encouraging. First of all, very importantly, it appeared to be well tolerated, generally well tolerated in patients with GRIN neurodevelopmental disorder with gain of function variants. The typical adverse events or safety observations that were seen were respiratory infections, which, as you know, are quite common in GRIN, or other signs and symptoms that were very typical of GRIN-related neurodevelopmental disorder. However, very importantly, there was a median reduction in countable motor seizures of 86% over the 8 weeks maintenance period. So a profound reduction over and above the seizure medications that the patients were receiving. Most patients had a greater than 50% reduction in countable motor seizures, and 6 of 7 patients were seizure-free for 80% of the days during the maintenance period. So really encouraging results in this study, acknowledging that this is an open label study, so there was no placebo involved, but nonetheless, these reductions were quite substantial, even in that type of study. Also, very interestingly, both clinicians and caregivers saw a clinical improvement in most patients on those CGIs that I mentioned earlier. And very interestingly, the improvement was seen regardless of whether they came in with countable motor seizures or without countable motor seizures. So overall, quite encouraging that maybe what we had intended to do, which was target the underlying activity in the disease, was actually happening. Again, the caveat being this is an open label study and must be studied in a controlled fashion, but certainly enough that supported advancing Radiprodil to the next phase of development as a potential treatment. And that's what we're here to talk about today. That's what we're here to really look forward and are excited to be initiating. It's a big transition when a drug goes from an early phase study like a phase 1 or 2 to a phase 3 registrational study. It means there's confidence in the company to invest, and there's confidence by the regulatory authorities to enable that transition. So that's where we are today. Next slide. Now, because of these data, we submitted to the FDA and requested something called Breakthrough Therapy Designation, and we received it. So this is a designation that FDA grants when that really is designed to help expedite actions that support development of drugs that are intended to treat a serious condition that's high unmet need. So it's not something that is granted for all drugs. It's just when you have a drug that has potential to really treat something that has to date been treated or not been treated or had been inadequately treated. What this means is that we will get fast track designation, meaning that this is something that the senior leadership at FDA and that the organization commits to help advance. Again, a very important step. And this was granted based on those profound seizure results that I shared with you with across GRIN genotypes. What I didn't share with you, we also saw this pretty consistently across GRIN patients, regardless of genotype, as long as they were gain of function. So all these factors contributed to the FDA saying, we want to give breakthrough therapy designation, and really does begin a regulatory partnership to help expedite development. Next. And this is what we're proposing to do. What we're proposing to do is to amend the existing study and have 2 cohorts once again in a phase 3 study. But in this case, there's a few important differences. First of all, the most important difference is that there'll be a placebo. And as I mentioned a few times, when you don't know if a drug works and when you're trying to understand the true safety profile, the gold standard is to use a placebo group. And this is always very difficult to know that there's the chance of being randomized to placebo. But it's truly the quickest way, honestly, to move the drug through development and come with the answer about whether it definitively works. And that's what we're proposing to do right now. Again, it's for all the GRIN types listed there with gain of function variants. There'll again be a countable motor seizure cohort, what we call the qualifying motor seizure cohort, and one where you can have less seizures or no seizures at all, but have a certain degree of neurodevelopmental or behavioral symptoms. The seizure cohort, patients will be treated for 12 weeks with placebo or Radiprodil, and then enter an open label extension phase. And for those without seizures, it's a 24-week maintenance phase. The idea here would be to have each cohort long enough to be able to see a potential benefit. The tools we use to measure signs and symptoms are what we call are not perfectly sensitive, if you will, meaning sometimes it just takes time to see change. You've all seen that. Over time, it's hard to see changes. The placebo helps that. The duration of the study helps that. But in each case, once the placebo control phase is complete, all subjects who've tolerated the drug will be eligible to receive open label Radiprodil up to the time of availability in the market, assuming obviously, that the drug is successful. So this is our current plan. We obviously, all of these plans in this business are subject to our colleagues at the FDA and the EMA, which is the European equivalent of the FDA, to help advise us and counsel us. And we're in the middle of that process right now. But there's broad support across the authorities that we met with to move Radiprodil forward. It's viewed as an important potential treatment, and the fact that we have breakthrough status actually shows the level of engagement and interest in doing the proper study and getting it done quickly. So with that, I'll turn it over to Hillary to talk a little bit more about the trial experience itself.
Thanks, Mike. So I'm Hillary Savoie. If you haven't seen my face before, I am the Director of Community Engagement and Communications at GRIN Therapeutics and Nervati Neurosciences. So Mike spoke a bit about some of the details of the proposed phase 3 B-line trial. And I'm going to walk through a few more details, some important details about who the key players are in a clinical trial and the steps that you all can take to get involved in research, clinical trials, and broader research as well. Next slide, please. So the first thing I want to talk a little bit about is the key stakeholders that are involved in a clinical trial. As we think about the upcoming pivotal trial and next steps, I just want to spend a few minutes here grounding ourselves in who all these people are, the terms that we use to describe them, and how they interact. Because oftentimes, you hear these terms tossed around: PI, site, sponsor, and even in some cases in this patient population, patient, family, caregiver. It's all a little bit confusing who we're talking about at any given moment. So let's begin by talking about who a sponsor is. So in the case of our study, the study that Mike just walked us through, Honeycomb and the proposed B-line study, the sponsor in that instance is GRIN Therapeutics. So we are the company that designs the clinical trial protocol and then all of the instructions that are involved in that trial. We are responsible for training sites to carry out that trial. So there are multiple sites, and we speak with all of them to make sure that they carry out the trial appropriately per the trial protocol and instructions. We work with advocacy groups like GRIN2B and GRIN to understand what the community needs are and get feedback throughout the entire process from them. And our other responsibility is to communicate with the sites in the study. When we talk about what sites are, and again, recognizing that this is a bit of a confusing term sometimes, a site is typically a hospital or some clinical setting that carries out the trial, that brings patients in to participate in the trial. And at a trial site, there is something called a principal investigator or a PI. And we're going to hear a bit more later from Dr. Kristen Park about what it's like to be a PI and what happens at trial sites. So I won't go into too much detail here, but high level, the role of the primary investigator or the trial site is to select and screen and consent patients. They are responsible for all of the medical decision-making for patients at their site, in the study at their site. So each site is responsible for the patients enrolled at their site. They're responsible for carrying out the trial as it's designed in the trial protocol and in the study instructions. And they communicate, as you can see with these arrows here, they communicate both with the study sponsor as well as directly with the patients in the study and their families and caregivers. Thinking about the responsibilities of being a patient or family or caregiver in a study. In this case, we're talking about a pediatric trial, of course, so it is the caregivers that take on the sort of role of patient responsibility. They're responsible for being informed and consenting to participate in a study. So there's a long process to involving an informed consent that they have to participate in order to join. They're responsible for following the instructions from the site as well as the study instructions, the trial instructions. They're responsible to understand what it means to join the trial and to make that decision to join the trial. They're responsible for respecting trial privacy. And so Mike spoke a little bit about the fact that in our upcoming proposed phase 3, there will be placebo control. And that's, one spot where trial privacy is really important, right? To not try to guess whether you're on placebo or on active drug for the validity of the data, right? For the trial. And they're responsible for communicating concerns directly to the site if there's a problem. So they reach out to the site, and you'll see there's no arrow between participants in the trial and the sponsor. So participants in the trial are not reaching out directly to the sponsor to express concern, but rather using the PI and the site to communicate with. They also communicate, of course, with patient advocacy groups. And as both Bruce and Mike alluded to, as a sponsor, we've worked really closely with the amazing advocacy groups that are in the GRIN space. And those advocacy groups have had a role leading up to this trial and helping design it. But they also have a role during a trial, right, to make sure that patients or their patient families are communicated to about clinical trials, what's available to families. They're responsible for understanding patient and family concerns, their needs, barriers to participation, and to communicate those issues to the sponsor. And we have had wonderful partners, again, in GRIN2B and CureGRIN in that process, and exceedingly grateful for that. The part that isn't shown on this slide actually is that broader community that isn't necessarily participating in a trial. However, I think we all know that rare disease communities don't really do anything in isolation. They do them in community. And so it's really important to understand that even if you aren't interested in or able to participate in a clinical trial, it's helpful for you to be aware of how trials work, who's involved, and why they work that way. Next slide, please. Thanks, Liz. So again, even if you're not planning in enrolling in a clinical trial or you're unable to participate, there are important steps that you can take either way, right? There are important steps that you can take to help support a better scientific understanding of GRIN-related neurodevelopmental disorder. So some next steps and ways that you can get involved. First, you can read more about the Honeycomb trial at UDRA-CT. The numbers at the end will help you be able to look up the exact trial and at clinicaltrials.gov. So that's just the European resource and the US resource. Again, that NCT number will help you search the trial. And right now, the information is about Honeycomb trial, but information on subsequent trials and other trials in the space are available there. You should stay tuned for updates coming later this month on next steps to learn more about the Beeline trial. We will have some updates coming. It's incredibly important to stay connected with advocacy organizations like GRIN2B and CureGRIN for updates on clinical trials and other research. They are our first to know and help disseminate that information to the broader community. And we absolutely encourage everyone to participate, again, regardless of whether you plan on being in a trial or not, to participate in efforts to better understand GRIN-related neurodevelopmental disorder, such as all of the registries that are available to you, any natural history participation, surveys, and so on. You can also learn more about us at GRIN Therapeutics on our website, grintherapeutics.com, or you can contact us at patientadvocacy@grintherapeutics.com, and that email comes to me, and I'm always happy to hear from families. Next slide, please. Okay, so just a thank you. I know both Bruce and Mike have expressed their gratitude. We are so, so grateful to our friends at GRIN and GRIN2B Foundation for being such great partners in this whole process and for the opportunity to speak here today. We also really want to take a moment to thank the scientists and collaborators and investigators who are involved in our studies. Certainly is not possible to do this work without a broad group of people. And it is absolutely not possible to do this work without participants in the clinical trials and all clinical trials. So we're very, very grateful to them. Thank you to the GRIN community for all of your encouragement and support, and a very happy GRIN Awareness Month. Thanks.
Thank you guys so much, Bruce, Hillary, and Mike. If anybody has any questions, you can stick them in the Q&A at the bottom, and we'll answer most of them at the very end of the presentation after we have Dr. Park on. But I do have 2 or 3 right now. We have about 10 minutes that we can dedicate to 2 or 3 different questions if you don't mind answering those right now. So I'm going to direct the first one to Hillary. We know that Radiprodil is for GRIN 1, 2A, 2B, 2D gain of function variants. What about patients who have not received a functional analysis, so they don't know, or they're inconclusive and were tested and didn't get an answer?
Okay, well, actually, I think maybe, Mike, if you want to start that off, and then we can.
Sure.
Yeah.
So I mean, we've been hearing about that. We know that that does happen. And we know that this is an area that has been actually evolving over time. I mean, even over the past few years, the progress on that process of variant characterization in terms of function has been increasing. We are actually putting a lot of resource behind trying to accelerate or facilitate that process. Don't have much to add to it now, just to say that because of the study, there's a lot of work that is going in and will be done on helping to facilitate that process. So I would just stay tuned to that. Again, check in from time to time on where we are, but we're working very, very hard to be able to facilitate that, because we know that that is a barrier and a burden for many families.
Awesome. That's perfect, Mike. Thank you. Well, Mike, you stay available because I figure the next couple questions you're going to have to answer anyway.
Yeah.
So one of the first ones is, were there specific mutations where the drug was more effective than others, and why do we think that?
There were only handful of patients in the study, so it's very difficult to say conclusive. Did one do better than the other? What we do know is that specifically when one looks at the seizure cohort, because that's the easiest cohort to see an effect, you see that regardless of genotype, whether it's GRIN 1, 2A, 2B, which are the ones we had in the study, the seizure effect was comparable, almost identical between them. So we know across GRIN genotypes that we are seeing a consistent effect. Now, across those genotypes, we have different variants within those genotypes. And so it's hard, that we just don't have enough to say whether there are going to be variants where there's less response. That work is those answers will come with continued clinical work, which is why, with the placebo-controlled study, we should be able to get at some of those questions.
Awesome. Thank you so much. Let's do 2 or 3 more. How does Radiprodil differ in its mechanism of action on the NMDA receptor from ketamine or memantine, and why start with Radiprodil first?
Well, I'll start on that, and then Bruce may want to add something to this. But I mean, ketamine and memantine do bind the NMDA receptor, but they are true blockers. They get in, and they have a blocking action, if you will, at the receptor itself. Radiprodil binds outside the pore and actually modulates the receptor, and by virtue of that binding site, is more likely to impact more variants just simply because of the location of the variants based on what we know about the NMDA receptor and where the variants occur in GRIN-related neurodevelopmental disorders. So I would say that, based on that mechanistic difference, that's really one important thing to keep in mind if one were to use one of the other NMDA blockers. But most importantly, the effects we're seeing here with Radiprodil in this population, those are data you're not going to find for other blockers. So, and we'll know for sure with the clinical trial. But Bruce, anything to add to that?
Just to key off of your chicken and fence analogy from the earlier pages, I'll just throw out a couple of additional analogies to help drive the point home. So when we think about antagonists like ketamine, like memantine, we characterize them as kind of a hatchet. When we think about Radiprodil, we characterize it as more of a scalpel. So it's more precise. It goes in and is able to do more fine-tuned, nuanced biological changes that drive clinical effect. And then another way to think about it is like a light switch. If you don't have a dimmer on the light switch, all you can do is turn it off or on. And an antagonist, an outright antagonist like memantine or ketamine, come in and turn it off. And it may not be best for patients to have it completely turned off. It may be best to use a dimmer and set the level of the light in the room to a level that's consistent with what you need to see, right? And so instead of coming in and turning off the lights, we're coming in and turning down the dimmer in these gain of function patients. And that's just more a nuanced, elegant way to modulate the receptor. So everything Mike says is completely spot on, of course, but these may be two helpful additional analogies to think about how Radiprodil is different from historical NMDA antagonists that aren't negative allosteric modulators. Think about negative allosteric modulators as the dimmer, and think about antagonists as the off switch.
All right, thank you so much, Bruce and Mike. I'm going to do one more question, then we'll move on to Dr. Park. Do you anticipate the trial could be impacted at all by the US's new administration?
So I'll start with that. I mean, I think that it's obviously there's a lot of uncertainty surrounding a variety of medical and healthcare functions. I would say we have had very good progress with the team at FDA, and they have been very reassuring that they're continuing working and they're continuing moving things forward. So at least so far, we have not seen an impact.
Great. Thank you so much. Okay, we're gonna switch over to Dr. Park now, and then we will come back to all these questions. So don't worry. Um, all right, let me add Dr. Park. There she is.
Hello. I
Hello.
got promoted to a panelist, my message said. So I said yes, but of course I will. Hi, good morning. I don't have any fancy slides, but I have had some discussions with Lauren and the team at Urbati about some topics that it might be helpful for me to cover. So I will introduce myself in case people don't know me, but I'm a pediatric epilepsy doc who does a lot of neurogenetics research and clinical care, and am the director of the Center of Excellence for GRIN Disorders at Children's Hospital Colorado, having been involved with the GRIN community since that initial conference at Emory. I am likely to be or will be a site PI, as Hillary mentioned in that structural chart of organizations. So someone at the University of Colorado, at Children's Colorado, conducting this upcoming clinical trial. And so wanted to kind of give some information about what patients and families can expect during that process. So, to start out, I will say that care inside of a research trial is very different from care in a clinical setting. Research trials are very rigorously controlled and monitored for safety, for processes that ensure the trial's data is going to be acceptable for the purposes that they set out to study. So that means there will be things called inclusion and exclusion criteria. So those are things that are about the patient or family, that says, you need to be able to accomplish these things. You need to be willing to come in and have these tests done, which could include blood draws or EEGs or other measures of safety, drug levels, things of that sort. You can be on these medications while you're in the trial. You can't be on these other medications while you're in the trial. You have to agree to tell us if something happens. All of these things are part of inclusion and exclusion criteria that determine if a patient is even eligible to participate. As Hillary and Mike mentioned, some of those inclusion and exclusion criteria have to do with what the symptoms are of the GRIN disorder. So how many seizures does someone have if they have epilepsy? And can those seizures be recognized by the family and counted accurately? And are there a certain number of them that occur within a specific time period? And so, when inquiring about it, the trial, you'll need to kind of determine if your child meets those requirements. And that is the responsibility also of the principal investigator and the team at the site that is conducting the research. Typically, the principal investigator is also accompanied by a research coordinator who is kind of the main point of contact for patients and families, who, helps with scheduling appointments, answers phone calls, gives you forms to fill out, and follows up if there are questions that you have. And so those people will look over, the information that you provide and say, I do think your child is eligible, or I don't think your child is eligible for the seizure cohort, but I do think they're eligible for the other cohort, or because of some factor, you're not eligible at all for participation. And go over that information. And then, so that's kind of at the beginning of the trial, kind of getting all of those things set up. And then there is a lot of paperwork in a clinical trial, a lot of paperwork. So the consent is usually very lengthy, and you want to make sure that you understand it. The person will go, either the trial, the research coordinator, or the PI, or both, will go over that with you. What does it mean to be in the trial? How often do you have to come to the center? What things might you be able to do over the phone? When do you contact the people in the clinical trial, and who should you contact and for what reason? All of those things should be very well laid out so that you will have an understanding of what it means to be involved in the trial. And then you sign that paperwork and begin the process that Hillary and Mike outlined of this screening phase, the dose titration phase, the maintenance phase. All of those different phases mean that things are happening at a different frequency. Certain testing is occurring at different times during each of those things. But most of the time, you are asked to kind of monitor your family member. How often are the seizures? Record them. How often are there problems with certain activities at home? Record them. So you have to really keep track of your family member during that clinical trial. One of the big jobs is for you to report anything that happens that's unusual. And that may happen in either arm of the study and with placebos. So if your child is hospitalized for seizures or, falls and breaks their leg, you have to report those things, even though some of them may be completely unrelated to whether they were or were not taking the medication. But it's the principal investigator and the study team's job to decide that. And that is very important if you think about when you look at medications and they say, there is a 20% chance that it would cause dizziness or a 10% chance that it would cause stomach upset. That's because people who are in the initial clinical trials reported those things during that trial, and they need to know how many times did it happen? Is it something that's common? Is it something that's not common? So that they can inform people later about taking the medication, what side effects they might expect. So even if it doesn't seem related, it's important to report those things to the clinical trial team, and they will ask you about them each time you come in for a visit. Has anything happened? Have you had any infections? Have you been in the hospital? Have you seen your doctor? All of those things will happen on a very regular basis throughout the trial. The other thing I think that's very different about a clinical trial is that you really can't make a lot of changes on your own in your child's clinical care. The trial is very regulated and constructed. So if you want to start a new medicine for seizures, you'll have to talk to your study team about that and whether or not it's allowed. If you want to decrease or increase the dose of a medicine that your child is taking, you will again need to talk to the study team to make sure that that is allowed. You shouldn't just do that on your own, like you might. You might have some leeway with your regular physician in clinical care to make changes in other organ systems. If your GI doctor tells you to do something, your neurologist might say, okay, well, I don't care about that. But in this case, they do, because you're on a clinical trial and everything is now regulated and monitored in a very specific and detailed way. So if there are changes that need to be made to your child's care, you need to let the study team know. So that is really important. I do want to echo, some of the points that Mike and Hillary made about participation and this mechanism. The FDA is a very conservative organization. Its biggest goal is to make sure that things are safe and for families and people to take them. And so it's very structured in how it looks at new medications and how they are approved. So the European Medical Agency, a little bit different. They're a little more flexible. They take sometimes different trial approaches, but the FDA has said we need a randomized clinical trial to authorize any new medicine for this space. And so, in addition, they also are typically much easier when it comes to things that involve epilepsy and seizures. The protocols are very well established, and they say, we know that pretty well. Certainly, the trial designs are not as great as they could be, and there are a lot of people working on trying to push the FDA to do better trial designs for epilepsy. But they have these standards, and they say we will accept them. We will do trials that way. And I think they're now being faced with a different perspective for these neurodevelopmental disorders that are genetically based and involve epilepsy, but also a lot of other symptoms. And they're being kind of pushed a little bit outside of their comfort zone in how to approach those and how to design clinical trials that will assess and act on a lot of the symptoms that patients and families are dealing with. And so I think it is great that we are pushing them and trying to design something a little bit novel by having this second arm that looks at the neurodevelopmental and behavioral symptoms in GRIN disorders. And the hope is that that will allow, a broader designation for this medication, in speaking back to some of the other questions about indeterminate, variants or patients who don't meet the qualifying criteria for this study. The hope is that when the medicine is approved, it will be able to be used not only for seizures but also for other symptoms, and that it could be used for patients, with other types of variants that are potentially gain of function. But for the purpose of the clinical trial, there really has to be, again, this eye on safety and efficacy for the trial itself. And so that is why really ones that are probably or likely gain of function are going to be included, and ones that are indeterminate or unclear are probably not, because they want to make sure that we can get through this first part with safety and efficacy in mind. So, I think that is a great step, and I'm looking forward to hopefully seeing how that turns out. I don't know. I don't know if I said anything that was out of order, or if Mike, you wanted to add anything, or Hillary. But I think that was my perspective on what it means to participate in a clinical trial. There's a lot of paperwork. There's a lot of responsibility. There's a lot of communication about things that may or may not happen, and questions about, okay, well, I'm coming on this day, but I have to get the blood draw today? Or it's very scripted, and so there is a little bit of flexibility, but a lot of it is going to be regulated during the period of enrollment. Sometimes things open up a little bit when you get to the open label extension phase, that things can be adjusted a little bit because that's a lot longer. But during the actual trial phase, things are very regulated.
Chris, the only thing I would add is I think the key word you've used about 4 or 5 times, regulated. It is
Yes.
it is heavily regulated. It is heavily regulated. It is, and I think the point of it not being clinical practice is another important thing that's very frustrating, disappointing. My regular doctor does this for me. Why can't you do this for me? There's a lot of people that get upset about that blame their doctor for that, blame us for that. This is how drugs get developed, and there are very strict rules on this, and there's very strict rules on this for the protection of patients and their families, because there was a bad behavior in the history of drug development, and that's what formed regulations, and it's for safety. And I always tell my team, it's amazing that any drug actually ever gets developed, because it is such a process. But it does, to cliche in a way, take a village, and every single member is important. And it is an amazing thing, I always say, that families are willing to commit of themselves to this process, because it is not easy and there are a lot of unknowns, but we're certainly grateful. And the only other thing I would say, put out there, is that part of this process is the trial design is developed with so much input from patients and also from our clinicians. So we try to do it in a way that is least tolerable, well tolerated, I guess, not just the drug, but the study itself. So
that was what I was going to add as well, Mike. I think it's really important. It's speaking from the perspective of someone who sat in a role like Liz and Lauren and my history. I think it's something that you don't always know how much effort is happening on the sponsor side to try to make sure that the design is as well tolerated, I like that term, Mike, as possible, and how much time is spent by our team trying to incorporate the things that we've learned from the advocates in the space, as well as clinicians in the space, to really action that feedback. There's a ton of work that happens behind the scenes in trying to do that. But it is still a heavy, heavy lift, and we recognize that. Very grateful for it.
Great. Thank you, Hillary, Mike, Bruce, and Kristen. So we have a lot of questions in the Q&A, really good questions. Lauren, I think, do you have an order, or I think you said you were consolidating.
Yeah, I'm trying to
There's
consolidate.
some
Do you
similar questions.
want to
Yeah.
add Russell to
Yes,
the
please.
Yes, let
Q&A?
me add Russell.
Thank you.
Thank you, Dr. Park, for everything you've done for us. Appreciative.
No problem.
Yeah, we have about 25 questions now, and
Oh boy.
I'm trying to consolidate a little bit. Um, Russell on real quick.
Yeah.
Yes.
Hi,
There he
Russell.
is.
Nice to see
Hi.
Hi,
you.
Russell.
Thanks. Been
Okay.
promoted.
All right, let's get started. Site locations, have any been confirmed yet?
So we're still in the process of site identification, and that's a process that requires that the sites have the capabilities, the resourcing to be able to do that. There's much that we can do to help sites create resources to enable participation, but there's only so much we can do. The FDA and the EMA are very strict about the level of expertise required at a given site to participate in a phase 3 study.
Thank you so much. Is Radiprodil currently available for off-label use?
No, Radiprodil is actually never been had been approved for an indication. I noticed another question in there somewhere. It has been tested in other disease areas. It was in clinical trials for diabetic peripheral neuropathy in the past. It had been originally considered at one point for other neurodegenerative diseases, but it is not available, but the safety data in adults is greater than 460 patients had been in healthy volunteers had received Radiprodil for short periods of time, even prior to it getting into our clinical trial.
Awesome. Thank you so much. Has there been any feedback from European studies yet?
So much of the honeycomb study was actually done in Europe, and so all along we've been very much engaged with the European regulatory authorities in the design of that study and the interpretation of the data. And the phase 3 trial that I've presented, right now the plan is to do that trial globally. So both the US and European regulators have been involved.
And Canada, I believe, as well, too. Yeah.
Yep, Canada and the Australians, I should say also.
Earlier, you mentioned keeping patients on their current medications through the study. What about memantine? Would the patient need to taper off that medication if they were in the study?
So memantine is one of what is known as an exclusion criteria, if you will. There are a couple drugs that for this patient safety or sort of interference with the action of Radiprodil, patients will have to not be on, and memantine is one of those drugs. There are only a few, but memantine is one of the drugs for the reasons we discussed earlier, given the potential to interfere with Radiprodil.
And I think that is part of the decision about whether to be in a clinical trial that people should have with their primary treating physician. Do you is it worthwhile for me to stop this medicine that my child may have been taking in order to participate in this new potential treatment for my child? And that's a very complicated decision that doesn't have a right answer, because there are no guarantees that that Radiprodil will be better, or that there won't be other side effects that will exist or will occur. And so I think that is something that has to be weighed by each family. If they're on an exclusionary medication, is it how effective has that medicine been? Are there side effects to that medicine? Is it worthwhile for my child to stop that medicine in order to have the opportunity to participate in another research trial?
Dr. Park brings up another very important point. Often with those drugs, there's a required washout period. So that would mean that that drug would not just have to be stopped and flip into a study. There's an observation period that has to happen to make sure the drug is cleared from the system with exclusionary drugs. And the other important part to keep in mind with a study like this, which is actually very common in these studies when we're talking about people on existing treatment, the other drugs that are involved for the treatment of seizures should not change during the study, at least for the maintenance part of the study. So that means you're also committing to staying on the existing treatment regimen, and just unless there's some sort of reason, like some sort of rescue, if you will, required. We're not going to let someone, obviously be at risk of seizure worsening. Obviously, if there's a rescue medication required, that can be used, but nonetheless, the commitment is to maintain stable therapy. And this is pretty typical of most studies. Dr. Park may want to add to that.
Yeah, and other therapies may need to be stable too. So we don't want kids to just be starting the ketogenic diet or just have a VNS that's placed, and people are still ramping up the settings for that. The goal of the study is to make the study medicine the only variable that changes during the course of the study, because that is the best way to assess how effective it is that it's the only thing that's changing, the only new thing that has been added. And so that's why there's so many regulations. It's, again, safety, the primary concern, but also efficacy. If you allow all these other things to change, in addition to the daily fluctuations of seizures and behavior in life, then it may wash out the effectiveness of the medication. So that's why there are so many restrictions on what can and can't happen. Now, again, that's obviously not to say that if a child develops a new condition, they can't be treated for that condition, but you still have to tell someone, and then that has to be put into the study data that says, oh, by the way, subject 35 developed reflux, and they had to start a new reflux medicine, so that when the rest of the data is analyzed, that can be factored in to whether that had any effect on the whole study. So it's not saying you can't treat things that happen. It's just saying that we have to know about it, and there may be regulations again about how it's done.
Yeah, to Kristen's point, we do ask that seizure management, whether it's ketogenic diet or medications, that be stable for at least a month before the screening process. So to her point about the need for controlling all these variables and making sure that we're just, as far as possible, looking at the effect of Radiprodil.
And I would just add that to come back to the initial question on memantine, how do we think about memantine, the royal we? We don't see it as interchangeable with Radiprodil. No physician would. And the rigor with which we're developing Radiprodil is just a very different approach to managing the underlying disease process than what's been tried empirically or, just with your best guess with memantine. So while the biology is somewhat similar, the NMDA receptors being targeted with both medicines, they're not interchangeable with our product candidate.
Thank you guys so much. Okay, let's move to how do you measure the improvement in a child who does not have seizures?
So right now the plan is to do similar to what we did in the honeycomb study, which is, we are doing a clinician global assessment, but we are developing a GRIN specific clinician global assessment impression CGI. That's never been done before. So we've done a survey of the community that you all helped us with to collect the information that was most important to families that have experienced GRIN-related neurodevelopmental disorder. We took that survey, and now we are developing a scale in which we're going to measure the impact of those important symptoms following Radiprodil treatment versus placebo. So the tool itself was developed with your input, and now we're formalizing that into an actual scale that can be used in our clinical trial. So that's a big development for the field. The other thing is that we're also now using other measures, like things like the Vineland-3, as well as the Aberrant Behavioral Community Checklist, which is actually other tools where we're using that survey in order to figure out what components of those tools we can also apply to measure the impact on things that are meaningful to patients and caregivers. And finally, we're doing interviews at the beginning of the study and at the end of the study to measure any change that we haven't captured using one of these more formalized assessments. So really, our overall objective is to try to assess, has Radiprodil in some way led to any sort of change that would be viewed as meaningful, even if the statistics might not sort of bear it out.
And Mike, if I might add just, my commentary on the next step slides about community participation in research. I think this is a really good example of, where participating in that survey, anyone who participated in that helped with this entire process, right? And I think it's so important that the community remember that leaning in and sharing your information, sharing your experiences in this way can really alter the path of drug development, and everybody who participated did that.
I would also add that, there is a lot of work being done on better and more detailed ways to assess children with neurodevelopmental disorders. However, that has not yet been translated to something that the FDA will accept. So a lot of these measures that the research study is using are measures that the FDA will accept. And they hopefully will be sensitive enough. I think I've heard, at least from a lot of my patients who do research, that the Vineland is like detested by families. But the fact of the matter is, it is accepted by the FDA, and other things that we may have access to or are working on are not. And so, we have to have this balance between something that is going to be sensitive enough to pick up improvements and changes over the course of the study, and something that is going to be acceptable to the FDA, who is ultimately the moderator of whether the medication gets approved or not. And so I think, while I would desire to have something, that's specific for GRIN or that's super detailed and so innovative, it's better to just do what the FDA says they will accept.
In our case, I would say our job is to try and make the detested questionnaires as tolerable as possible.
Yes.
So the way we're doing that is we have a central administrator who's going to basically allow scheduling of it at the parent's convenience, rather than having it done with the 10 other tests being done at the center during the actual very stressful dosing visits that often come with a trial. So we've heard that how many of pretty much all tools are detested. They're very painful. They take time. Not painful pain, but just stressful. We're trying to adapt as best we can. And the GRIN specific elements here are the CGI, which we were very pleased that the authorities are supportive of us developing. And we've been working. Also, we're going to be looking at the ORCA, which we know the community has also been involved in trying to bridge from the work done in other neurodevelopmental disorders to GRIN.
And just as a final overarching comment on non-seizure outcome measures and the path that we've been able to chart here, it's, to Mike's earlier point, a very positive commentary that we've had the kind of constructive engagement with regulatory authorities that we've had in order to pursue this sort of path. And it isn't easy to find ourselves in this situation. So it's a very positive commentary on the level of constructiveness that's being brought to bear on this study, and the underlying biology, recognizing that the audience today may not necessarily have been trained in science or clinical medicine. We use words like biology, and it may seem abstract, but it's actually quite specific. And the way in which we're aiming to target this disease and to target its underlying cause, which is underlying biology, has resonated with our regulatory colleagues in such a way that has enabled us to chart a course to look beyond just seizure. And that's a remarkable accomplishment for us, for you, for patients. It's representative of the kind of I'll use the same word, constructive approach that everyone that's a part of this is taking to realize the maximum benefit for patients.
Well, thank you all so much. Next question. What is the patient selection criteria, especially age, for the phase 3 study?
I'll take that. Based on what we know now, we're looking at patients as low as 1 month of age up to 18 years. So we're trying to have pediatric into adulthood.
I realize that GRIN is quite rare, but do you feel that a sample of 12 or 15 proves safety efficacy?
I don't think we can claim that the sample of 12 to 15 proves safety or efficacy. That's really what the purpose of the phase 3 study is, and the addition of placebo. We need to understand, this is a complex disorder. Clinical trials are complex. They're associated with a lot of biases. People want clinical trials to work. Therefore, you need to have this control group, which is the hardest part of a trial, but we're all biased, especially people who want to see their patients and their children do better. So that's why we're doing this.
And I would say that even the end of this phase 3 trial, will hopefully prove safety and some amount of efficacy, but the ongoing work, there will be ongoing work that will define that further, right? So and that's, I think, the work of the GRIN research community is to then subsequently say, okay, well, hopefully after this medicine reaches the market, now my center has seen 100 patients and put them on it. This center in the Netherlands has seen another 100 patients. Let's put those 200 patients together and see if we can learn anything more. Like, hey, it worked best for this subset of people that maybe we only had a few of in the research trial, but now we have enough to say, oh, specifically, it's really beneficial for those people. Or, yeah, there's this other subset where, yeah, maybe that's why they didn't have a great response in the research trial, is because of this particular thing. And we're going to try to characterize that further, right? And I think we do want to continue to hear about families who take this medicine and learn about other things that, that happen in the long term, so that we can, again, make better and better decisions about who to prescribe it for when we have the option.
Yeah, and as we accrue more natural history data alongside the Radiprodil data, that will be very important to help us understand the safety and efficacy.
I think when companies develop medicines, this is not a comment that's specific to GRIN Therapeutics. This is an overarching comment for all drug development. Safety is always of paramount importance. And if you are drug developers trying to determine the safety profile of an experimental therapeutic, you are highly focused on safety. Mike made that point earlier. One of the features of this particular molecule, not in GRIN-related NDD or even pediatric patients for that matter, but in humans, meaning adult humans, we have a fair amount more safety data than most medicine or therapeutic candidates do have at this stage of their development. And that was a feature of Radiprodil that gave our backers significant comfort, because there were literally hundreds of humans that have been dosed with this medicine, I should say therapeutic candidate. And that was helpful to us in trying to understand what the profile of the drug might be in this particular patient population. As I mentioned, it's not several hundred patients of GRIN-related neurodevelopmental disorder data, and it's largely adult data, but it is still humans. And it gives us a significant amount of comfort that what we're able to organize around in terms of safety profile or potential safety profile exceeds what folks are ordinarily able to organize around when developing experimental therapeutics.
Thank y'all so much. What is the likely timeline for Radiprodil being fully approved by the FDA?
Well, I mean, the best I can tell you is that, let's get the final study design down. We'll be in a better position to do that. It is driven by how fast we can recruit the study. And that's really going to be the rate limiting step, is how quickly we can get, as you saw on the slide. You may have noticed it's 40 or so patients in each group. That's 80 people with GRIN gain of function variants. That's going to take some time. So that's going to be the main driver of that. And that's where the community can be very helpful.
Will the placebo control group know their status as such at the end of the trial?
After the trial, generally the way this works is they'll go on to open label drugs. So if you've been on placebo for 12 weeks or 24 weeks and you still want to stay in the study, you get Radiprodil. You'll get a Radiprodil and you'll stay on Radiprodil. After the study is done, meaning locked down everything, then patients and families will be able to know, but not until it's done. But that doesn't mean you have to wait to get Radiprodil. You're going to get Radiprodil as soon as the placebo period is over. So you don't have to wait.
Were any duplications tested?
Duplications. I don't know if we know that. I'm not quite sure. I don't think so.
I think we limited to missense
Yeah, I think so.
mutations.
Yeah, right.
So,
You're right.
yeah.
Good point.
Okay, is Radiprodil currently FDA approved for other indications? And also, is it available for use in vitro assays?
Not in other indications. And we're generally not giving Radiprodil for basic science use at this time, now that it's in phase 3.
If phase 3 has similar results to the previous phases, will the drug be approved for use or will there be another trial?
I would say if we see 86% reduction in seizures and a benefit on behavior, we won't need another trial. We've
Yeah.
actually, I think it's the rare disease here. If you have definitive effects like we've seen in a placebo-controlled way in a rare disease, I don't think there would need to be another trial for this. Will there be other trials in other disease areas because of the potential? Absolutely. We're already doing that in tuberous sclerosis complex and focal cortical dysplasia type 2. But for this, no. And
Sorry.
Go ahead, Chris.
I was just going to say, so for some context, other drugs that go for approval for seizures have a 60% response rate. And Onfi, I think, is the one that has the highest response rate currently of about 70%. So having an 86% reduction in seizures is quite a bit different and quite a bit better. I think obviously hard to draw any of the behavioral indications, but when people have used the word pivotal, what that means is that the study was designed specifically with the aim that if it had a positive result, then it would be sufficient to allow the FDA to approve it. That's how all medicines, they have these designs that they work with the FDA and they say, okay, if we do it this way and we get your threshold effect, will you approve the medication or the therapy or whatever? And they say yes. And so that's what's called a pivotal trial, means that it's designed that way. And if it has the anticipated benefit, then you just hand it to the FDA and they review it and they say, okay, thanks for getting that done. I mean, obviously it's more complicated than that, but essentially that's the role, that's the design. It's designed that way so that it's going to give them the information that they want.
Yeah, I wonder if with the question related to the idea whether we need two positive phase 3 trials in this rare disease.
Interesting.
In this setting, we don't think that's going to be.
Yeah, Mike, I wonder if it might be helpful for you to just say a few words about why in rare indications you can alter the course of development in conjunction with regulators.
So yeah, I mean, there are all these different ways of developing a drug, and in rare disease, sometimes you can combine the phases. Like we're going phase 1B, phase 3. That's very unusual. Often they'll do a phase 2 with a phase 3, so you can combine things. And there's more flexibility in the setting of a rare disease, especially when there's no treatments. I mean, that's what breakthrough designation indicates, the potential to have something that is different than other drugs. In those settings, global regulatory authorities do generally allow you a bit more flexibility. So,
And To the question, sorry.
Go ahead.
To the question that came before about duration, I think the ability to push the phasing together in this way, while we don't know how long everything will take and it's subject to enrollment in the trial, it does have the hopeful effect of moving more quickly, right, to
Absolutely.
Are we doing orphan drug designation or no?
So we will be, that is something that is part of this. This is an orphan disease, and that's part of the process that we're going to explore. But yeah, so we're gonna sort of avail ourselves of all these different aspects now that we have a little preliminary data. That helps. When you have hopeful data, it helps. I know do we have 12 questions left? Is that still in the next 3 minutes, 5 minutes? I just want to make sure.
I'm trying to combine them as best I can.
Okay, perfect.
I'm gonna try to combine this one. So aside from seizure reduction, people are wondering about cognitive observations and improvements. So what other symptoms, including cognitive, were labeled and tracked, and are there any specific percentages of each?
So we saw trends in the CGI and the CAGCI, as I mentioned, and we also saw some impact on the ABCC in the phase 1B study, so the honeycomb study. So that's the preliminary information we have, and that's what we're using to design the B-line. They're using the same endpoints, if that answers the question.
When could we see Radiprodil available to the general public if the study is successful?
So I think that goes to the previous question of as soon as we can get it developed. The good news about this is, as Hillary was pointing out, we have the flexibility of combining phases. We have the breakthrough designation that gets us fast track, and that also gets us what's known as a priority review, which means that shortens the FDA's timeline from 15 months to 8 months when it gets time to review. So that all of those things help, but it still takes time and is completely driven by recruitment.
Great. Is GRIN Therapeutics doing any research on loss of function?
We've thought a lot about that, we are actively considering approaches to loss of function.
Awesome. Is Vicompa on the exclusion list?
Is Vicompa on the exclusion list? Yes, it is.
Yeah, that's perampanel. Yeah.
Perampanel. Yeah. Yes, it is.
Okay. We feel totally left out here in Africa, especially South Africa. No locations in the last phase, and also such time lags between FDA approvals and then distribution in the third world. How are the therapeutic companies addressing these issues?
Well, I can start, and I don't know, Bruce, if you want to add anything to this from a broader perspective. I mean, I think that we're open to going to where patients are as best we can within the regulatory rules. So I think that the goal for us would be to be able to get the Radiprodil to the places where it needs to get to for both the trial and to address patients' needs. That's the eventual goal. We're just getting started as a company. We're just getting started as an organization. It's not unusual for a phase 1B study not to be in many countries. It does change dramatically for phase 3, and it absolutely changes for broader exploration and eventually commercialization. We haven't decided where we're going to be from a global study perspective, but the goal, again, we'll go where we have to go, both for the trial and for commercialization. I don't know, Bruce, if there's anything you want to add.
I would say, as we think into the future, should we succeed in the pivotal study, the phase 3 study, our intent is absolutely to be across the globe, making the medicine available, if approved, to treat any patient anywhere with GRIN-related neurodevelopmental disorder. Mike's points are related to the clinical trials work that we've done, and those points are spot on. But even though the studies may not be conducted in certain parts of the world, it is absolutely our intent, and there's work ongoing right now in the background to enable broad global distribution should we achieve approval.
Thank you so much. Do you guys anticipate that Radiprodil will have long-term efficacy?
We, so yes, I mean, we're doing that. Everybody who was treated in the phase 1b study is now in a long-term efficacy study, and we have people who've been there well over in that study for over a year. So we are continuing that is a requirement, by the way, to show long-term effects as part of the regulatory process. So we're actively pursuing that, and we haven't seen any indication to date that there's a wearing off effect, as can happen sometimes with drugs.
Awesome. Okay.
But I think that's still a bit of an unknown. I mean, I saw the questions, in that led up to that, which were about various seizure medicines and other types of specific GRIN therapies, kind of working for a while and then kind of no longer working. And I think that is a lot of a lack of understanding in the neurologic community about why we have that pattern especially in intractable epilepsy, where seizures remit for a while and then they come back. What are the mechanisms within the brain that allow that to happen? I think they're still kind of unknown. And so it's hard to say we figured that out. And so we know that Radiprodil is going to be able to bypass all of that and last longer than other things. So I think that's still hard to say, but I think the hope is that when these things are more specific and more targeted, that they will do better in the long term, but I don't know that we can really guarantee that.
Lauren, I know we're at time, but there's one additional question that I don't believe we've covered that if you don't mind me just jumping in and
Do it.
answering.
Go ahead, Bruce.
So there's a question on whether or not we would expect Radiprodil to help patients with GRIN2A mutations, given the receptor that we're modulating, the subunit that we're modulating is the NR2B subunit or GLUN2B subunit. And the answer to that question is yes, we would expect. It is our hope that this medicine therapeutic candidate would work in all patients with GRIN-related NDD. Certainly, the data we've generated to date demonstrates that independent of the gene that's affected, there's impact, positive impact. And that's because of the way in which these receptors are created in the brain. There are multiple different subunits that go in to create the full receptor. And those subunits would come together to create function at the receptor. Our ability to modulate GLUN2B subunit has impact on other genotypes because they're all coming together to form this multi-unit, multi-subunit receptor. So important for folks to understand that while we're targeting the GLUN2B subunit, we've seen impact in different genotypes, and our hope is that we'll continue to see that impact, and the biology would support seeing impact across multiple different genotypes.
Awesome. Thank you so much for that. And I think, Russell, are you answering the last question on there?
See Russell doing it. Go ahead, Russell.
Thank you so
I just did
much.
that.
Go ahead.
Yay.
Done.
I can't believe we answered all those questions basically at time. Great.
I
Very
saw a
impressive.
few. I know it's like time, but I saw a few other ones. I think, is this presentation going to be available, Liz, for people to review on the website? What should they do if there are more questions that maybe they didn't get answered or things of that sort? What are the options? I think Hillary gave, her contact information, but I'm not sure if that's the best way to go. What are our thoughts about that?
That is a good question.
Something real quick and say that this
yeah.
has been recorded and I will upload it to our YouTube channel, GRIN,
Great.
GRIN2B. We're going to upload those for you guys to view later. But Hillary, as far as contacting, I'm assuming your contact information.
If there are questions that weren't answered, you can use the patient advocacy email. Also, Liz, Lauren, if you are collecting a set of questions that didn't get answered, you can pass them directly to me as well.
Great.
Yeah.
Awesome. Well, thank y'all so much. Thank you, Liz.
Yeah, thank you.
Thank you.
Thank you, everyone.
Thanks so
And
much.
thank you for doing this during GRIN Awareness Month. This is very exciting.
Thank you for inviting us.
Thank you very much for all of your engagement.
All
Y'all
right.
have a
Bye.
great day.
Okay.
Thank
Bye,
you,
everyone.
everyone.
Thank you.
Bye-bye.
Bye.